Welcome to the wonderful world of mitosis. Mitosis is also called cell division or cell reproduction. It is a process that happens when we need some new cells. Perhaps some are dead and dying, some are old, some have been damaged, or maybe we've cut something and we need to heal it. Maybe we just straight up need more cells to grow, but this is when mitosis will happen. Today what I'm going to show you is a model of that process called mitosis. What you see in front of you is a whiteboard which you can get in class. You can see some pipe cleaners that we use as a model of our chromosomes. You should have 4 different sets, two that are long and different colored, 2 that are short and different colored, and that's the way you should always begin. Now to understand the process, I'm going to show you the steps in the order that they come into. The first step is called interphase. Interphase is where we have our normal looking cell. Now, if we can imagine the edge of our whiteboard as being the cell boundary, whether it's a cell membrane or the cell wall, but we're looking at that as our cell boundary, our cell barrier. That's going to be given on the outside. I'm going to draw it in right now, but eventually that will probably change and go away, and we'll have to kind of envision that a little bit. But we start off with the nucleus, and we start with our membrane intact. When we look at this, this is interphase. Interphase is the longest phase that we have, and we really, in our nucleus, we don't see a lot if we're able to look at it. Right now, the DNA is all stretched out, and the genes are being read and that kind of stuff. At the point where we're going to trigger mitosis to happen, the chromosomes or the DNA will go through a process called replication, where we'll be seeing the original strands of DNA making a copy of themselves. We call that DNA replication. So in interphase, the big thing that you'll have to know is that DNA is indeed replicated, and we're going to see this in our model as these extra chromosomes being created. Now, can we really see that? No. What we're going to see is really the nucleus as being solid, but that's because the DNA is all over the place, like kind of a big bowl of spaghetti. This is happening even though we can't see it happen. That's interphase. Interphase is the longest phase. It's there the longest, and then it goes into something called prophase. Okay, so from interphase we go into prophase. Interphase, we have the chromosomes replicated. We can't see any of the chromosomes at this point. In prophase, there's a few steps that we're gonna take. One is that our nucleus is going to eventually completely disappear, and that's going to happen probably throughout the prophase, but I'm going to take it away right now so I don't forget, but it does disappear. That allows for the chromosomes to actually get moved and pulled apart into different parts of the cell without the boundary called this nuclear membrane. So the nucleus is going to disappear. Now we have these replicated chromosomes, which we call chromatids. You see the 2 purple ones. We have 2 chromatids here. We call them sister chromatids more specifically. So here's a sister chromatid, here's a sister chromatid, and down the line here. The second thing that really happens in prophase, or maybe while the nucleus is disappearing, is that these chromatids are becoming chromosomes. So they're going to start to coil up. Whereas in interphase we couldn't see them, the prophase chromatids we can see because they're starting to coil up and become thicker. While they're doing that, they will find their sister and they will pair up. So we call the sister chromatids pair up and now can be seen, and we see them as these X structures, which we call the chromosome. The chromosomes are held together at the center of the X by something called the centromere. The last thing that really happens in prophase is going to be the appearance and then the duplication of the centrioles. The centrioles are the organizing centers for cell division. Now, normally you'll see them appear, duplicate, and then they're going to move to the pole. So I'm going to draw them in at the poles of the cell right here. So those are my centrioles. Okay, so now we go from prophase into metaphase. So we've gone interphase into prophase now goes right into metaphase. Now there's no stop to this point, so what we're seeing is kind of snapshots along the way, but hopefully we'll get this all put together so it looks like it does in the cell. In metaphase, these chromosomes line up right along the middle of the cell. You'll see them line up along this middle line of the cell called the metaphase plate. And as they line up along the middle, these strings from the centrioles appear. We call these spindle fibers. And these spindle fibers will connect to each of the centromeres. And so, and those spindle fibers will make sure that they are in the center and that they all get lined up, and there's some organization to this, this phase. Okay, so from metaphase, or where they line up across the middle and meta, now we go into anaphase, ana or apart. So we see the first letters match up with what was really happening. In anaphase, the centrioles and the spindle fibers will pull on these centromeres and break the sister chromatids apart from each other. They're going to pull them to those poles. And they're going to pull right out to the centrioles. And as you do that, you'll see these spindle fibers shrink up into the, back into the centriole. Okay, so now at the end of anaphase, we have a group of the chromosomes or the chromatids over here and a group over here. And finally, we end up at telophase. So we've went from interphase into prophase, prophase flows into metaphase, metaphase flows into anaphase, and finally we have telophase. In telophase, a couple of the main things that happen here is that we have our nucleus reappear. And our nucleus reappears, so our nucleus is going to reform around each of the groups of chromosomes. The cell itself is going to start to pinch in, and we're going to end up dividing down where that metaphase plate was, where all those chromosomes lined up, but we'll have this pinching in process, so it'll pinch in at the edges and then it'll split along the middle. We call that cytokinesis. Cytokinesis, cell moving apart, so we'll start to see these cells split and then move apart. So we'll have a cell here and a cell here. Each are going to be exact copies of the original cell that we started off with, especially when we start looking at the chromosomes. Notice that we have one lime green, an orange, a purple, and a blue here. That's exactly the one we started with, and same goes for the one over here. So at the end of telophase, we have two brand new cells created from the first cell that we started with, and they're identical copies of the first cell. So again, that would help with us if we need new cells. We want the same type of cell. If we are going to try to heal, we're going to want the same type of cell where we're healing at. If we're growing, we want the same type of growth cell. So again, we have mitosis creating of this at the end of telophase, and the splitting of those cells, which was called cytokinesis.